Hello, my name is Leho Tedersoo, and today I'm showing how to describe new species and higher level taxa based on DNA and sequence data. So I'm sharing my screen with you, and let's go through the various steps. So I have prepared guidelines for descriptions. So first of all, you need specific software besides Excel. You would need AliView for alignment, PicPic for screenshots and cropping, and Figtree for viewing tree images. You will be supplied with the following files. First of all, an alignment file of all eukaryotes. It looks like this. So here the model organism Saccharomyces cerevisiae is on the top, and other taxa are following. Another alignment file is alignment of the target taxon. So today we included 107b as an example. So these taxa are aligned against Saccharomyces cerevisiae. Then we have also an Excel template file which includes metadata and a template for Word for entering text. Here you will need to fill in or replace uppercase words or abbreviation as 666, YYY, and ZZZ, but not the question marks, because for this you don't have enough information. We also have a tree file of all eukaryotes containing all taxonomic groups. It looks like this. So the target taxon 107b is indicated here in the yellow, bright genus level, and also family level and phylum level groups. So you can also see that this taxon belongs to this Ophistogont group that also includes fungi, animals, and related protists. This tree you need to get the sequence accessions for particular target taxa. There is also another tree about this particular group. Here is the genus or species, the whole genus, family, and also the rest of the phylum, which is quite distinct and has a long branch. The metadata template looks like this. It's sorted by group. First is the type species and type sequence, and substrate, country, locality, geographical coordinates, and hidden are also a few extra fields of information about the biome and dominant vegetation in the site, which is not important here. So one important thing to know is also the ribosomal RNA genes, because we need to map the diagnostic mini barcodes according to localities. So first, the Saccharomyces cerevisiae starts exactly from the first position of SSU, and it ends in position 1800 with this ending, CATTA. The 5.8 gene start is indicated here. 5.8 as end indicated here, and start of large subunit is here with all these positions. For the entire work, I have also prepared rest of the instructions. I won't stop here in details. And there is also the template with abundant commentaries about what to fill in specifically. We also developed a system where the person preparing the diagnosis will be the name author for phylum, class, order, and family, whereas the proposer of the genus name will be the author of the genus, and proposer of the epithet will be the main author of the species. So in this example, let's start with this our defined group. So it should be 107B, which we agreed or voted that is the species is Taphomonas livonica. So the phylum name would be Edaphonadea, and the authors will be according to what we proposed. It's always easier to start from the smallest rank. It may be a bit strange to start from the bottom, but that's the way it is. And we can first start with the etymology. I have copied this etymology information from the voting file to here. So let's fill it in. Taphomonas in Greek refers to a flagellate organism from the soil. Actually, unicellular organism from the soil, which is obviously related to its habitat. This is based on the metadata file here. You see that in most cases it is indeed soil. This has not been previously recognized group, so we delete accession and reference part, and the epithet livonica refers to the locality. Livonia means Estonia and northern Latvia, where all findings of this particular species occur. So findings in Estonia and northern Latvia. historically known as Livonia. Again, no other accession number or reference, so we delete this part. Let's use this information also for the name. So copy Edaphomonas to the genus name and Livonica epithet to the species name. Livonica was proposed by myself, and that's why I will become the author of the epithet, but also Enrique was involved in this description of the group, so he will be in the second position. Also in bold font as a subtitle and into italics. Later we will have to submit it to Zoobank, so we don't know this code. So let's now go to the type material. We have the type sequence, which is this one from Eukaryon. So let's copy it here in the template. It goes to this sequence field, and to question marks we will later add the INST or NCBI code, as we will of course submit these sequences to GenBank as well. We use preferably the physical soil sample as a holotype. So it is the global soil plot number G4785, and we add this here. Later we will substitute it with the voucher name. The eDNA sample is the same name. We have it also vouchered, so it will also get the additional number. So it's this global soil microbiome plot. It's number the same. It has been found in soil in Estonia, Mäe village, and in temperate forests dominated by Acer platanoides. So we can add this information here. Soil in Acer platanoides forest in Mäe, Estonia, and we also copy the coordinates to here. We preferably use 4 basic numbers, and we should round it upwards. So this 4th position is indicates 10-meter precision. The longitude as well, and now we are principally done with the type. So in the description part we will include other sequences. So these are displayed all here. In total there are 7 others of them. and we will copy them here one by one. We are doing this in a random order. If we later find some other reason to change the order, we can easily copy them in the back or forward positions. So the remaining we do not need. So let's go through the first of them. Others I can finish later because it will take some time to add and to double check. So the second in the list of this comes from a moss sample in a small city called Paldiski in Estonia, and the moss has grown on a living tree. Okay, so the plot is it's actually the global soil plot, although it's not a soil sample, but we can add this information here still. So habitat is moss in Paldiski, Estonia. And we copy also the information about coordinates again. So others I will fill later, taking more time and care. So next we will go to the diagnosis. Here it's critical to find those micro barcodes to distinguish this particular species from other congeners. There are quite a number of them, altogether 28. So let's look at the alignment. So the alignment file is here. It's outgrouped by Saccharomyces again, and this type sequence of Etaphomonas livonica is long enough to reach in the very beginning of a small subunit. It's an exception. Usually this is not the case. Mostly the sequences start from around 600 bases, where variable region 3 starts. So this is related to the primers that we have been using. Also important about AliView is to put the settings right, because we need to take screenshots. So here we have to tag that always display uppercase letters, so we can get uppercase also for the screenshot. And for colors, we have to define nucleotide not as default, but as CView. So we get those nice bright colors as we see here. In general, I recommend to start from the ITS region. So let's find it. We quickly viewed what are the signals, starting signals for those different regions. So if you carefully take a look, then okay, see this SATA ending for Saccharomyces is here. The Taphomonas is also well aligned here, so it's in the same position. Second, let's find start of 5.8s. Yeah, all right. So in Saccharomyces, it starts from here, and we see that Taphomonas is very much distinct in beginning from Saccharomyces sequence, so maybe difficult to recognize without alignment. And when it comes to the end, then I see the signal here. So this is the very end of Saccharomyces 5.8s. So next there will be LSU. After this ITS2, LSU is aligned here, and LSU starts with this signal here. So in this case, it's all well defined because they align actually well. So the first task is to find some kind of discriminating region either in 5.8s or ITS2 in this region. So let's go back. Okay, so here is the 5.8s, and we just have to start comparing our target taxon with other neighboring species. You see that in 5.8s they are very similar, but in ITS2 there start to be groups that differ from our target species, but still some other species may be very similar in their sequence. So here we see that it's not very easy task because the sequences are either very different or very similar, and in some cases there are also single nucleotide differences within the target species itself. So these places we have to ignore because these are not suitable for our purpose. So let's try to find this place in some most variable region. For example, this chunk stands out. Our species is here. There is nothing else with position C here, and there are some other strong position like another C here, and also A and G here. When we check the length of this, right, it's exactly 20 nucleotides, so it's well suiting for our purpose. If it's too short, we could add extra material from either right or left, however we feel appropriate. So let's first take a copy of this suitable region. We can paste them in an Excel file. And now we also want to know what is the position of this first nucleotide, because we have to map it. So carefully we go back to this alignment. So here goes the end of 5.8s. So we take the next first ITS2 nucleotide. We copy it with caps with Ctrl+C and paste it here. For ITS2, it doesn't make sense to include Saccharomyces control because ITS2 is not alignable. So we refer only to this particular type. So here we will remove caps. This can be easily done just by replacing the dashes to nothing. So now it's ungapped, and using the len function in Excel, we can simply count all the nucleotides. So indeed, for the microbarcode, it's 20, and it appears that it's 76 nucleotides to the first nucleotide of our microbarcode. So it means that our microbarcode starts at position 76 in ITS2. So we copy from Excel this microbarcode to the text file. Let's find where is the ITS2. ITS2 is here. This YYY is meant for this location. Based on the alignment, we can also define how many mismatches we allow so that our species would still be recognizable amongst others, knowing that ITS2 is quite variable and other species were at least 3 nucleotides distinct. We can allow no or one mismatch. So let's allow one single mismatch. So the position was from 76 to 95 in this type sequence. This is what we call legit type here. And one of our tasks is also to specify what are the particular distinguishing nucleotides in this mini barcode. So for this, let's go back here. Okay, it seems I lost it. Yes, we measured this length, so it was lost for a moment. Okay, so here we can see again that this C is unique. The next T, G, A, G, T are not. And then this third G also tends to be relatively uncommon among others. So we can underline these two already with Ctrl+U to indicate the underline. So A is not unique. T's are not unique. Then again this C. And the terminal A and G are unique. So C, A, and G. So roughly 6 well distinguishing nucleotides to other taxa in this group, this genus. Of course, they are not all exclusive differences at all. So the next step is to take a snapshot photograph of this. So I request that we zoom in so that there will be exactly 34 sequences or entries in the screen. Now it's 46. So this is to make the quality of the analysis or the screenshots comparable for publication purpose. 38. So another round of zoom. And now it's 34, as we see on the bottom. So the idea is to include 10, but not more, of the entries of the species, starting with the type. So here we have 8, so we keep all of them. And we select a number of congeneric species also for comparison. So here seems to be a nice bunch, and they are already aligned. If they would be scattered, we could a bit help to fit the alignment better into the screen. This would be just for the visualizing purpose. So let's move this target region to the left and leaving some space. Then we open PicPic. Here we have already one example from yesterday's effort with some extra margin and including the entire species information. So starting a new image is easy. Click on the region and select with some extra margin all of the particular mini barcode. So later we can trim this further, and all this text in the left will be manually inputted again in Corel, so that the format and size will all be the same. We save it as a PNG file. It's region 107p, and it was the ITS region. So we save it in the screenshots folder. So now we are principally done with the ITS, and next we can go for the small subunit. So let's zoom again for better visibility. For the small subunit, I recommend to start from this variable regions 3 and 4, because these are the most commonly used. And here, our long reads also start. So in this starting area, we have 3 sequences from Metaphomonas livonica, and we have also 6 sequences from the Etaphomonas genus. And we also have a number of sequences from this larger family. So here again, the main idea is to find some positions where Metaphomonas livonica can be distinguished from other members of the genus and also other members of the family, because it has to be pretty unique. So when we compare, we can actually drag these informative sequences here closer to the top and also the family, so it will be easier to compare. So we see that right here in the beginning we have one place where Edaphomonas livonica has some unique sequences. So A is here, also white unique T is here, and also some very unique A nucleotides here. So this might be a suitable place. We can look for alternatives as well. When we scroll to the right, it becomes a very conserved region. No unique differences. Here is a very long intron, characteristic to the other members of the family. That's indeed really long compared to regular introns. Yeah, again, we don't see anything unique, and we are getting quite far to the right as well. So since the first one looks fine, we can use this one. So let's double check. We can start from these nucleotides. Although they are not unique, it's possible they are not unique, but compared to several others conspecifics, there is a 3T insertion in these others. So if we realign it here, we can actually make it more distinct to some other members of the genus. So this feature is shared by 2 other members close to Edaphomonas livonica, but these can be distinguished by C nucleotide in the 5th position, and one of them also by A nucleotide in the 6th position. And additionally, there are 2 A nucleotides that are partly distinguishable and one A nucleotide that is also fully distinguishing. So this is 19 nucleotides. Our preferred standard is 20. So we can take this part just for clarity. We can take this other part away so that we can focus only onto this. We also see that the Saccharomyces alignment is matching, so we don't have to worry about these exact positions. And here we can refer to both Saccharomyces and the type sequence because they start in the very early in SSU, from the very first nucleotide of SSU. So again, we copy the sequence of the type, which is 20 nucleotides. We copy it to the same table. Okay. It seems to be auto-saving in a wrong time. So let's wait a little bit. So auto-saving done, and we save our stuff. So next we compare it to we map to the position in the type sequence. So we copy this one to Excel file. So yeah, and in addition, we will also refer to the Saccharomyces cerevisiae. Again, to the very first position. So replacing caps to nothing. And dragging the function also to cover Saccharomyces cerevisiae. So we see that the microbarcode starts at position 667 and in Saccharomyces in position 642. So let's add in this information to the text. First, microbarcode. SSU. The field ZZZ. Since SSU is very conserved, also the variable regions, we certainly allow no single mismatches. There could be if there are some sequencing errors, of course, but it's also likely that it's another species if there are some mutations in that region. So position 667 in the type. 667, it was 20 nucleotides, so 686. And 642 to 661. Again, let's determine the very characteristic differences. So in general, the second, third, and fourth are quite distinct, not entirely, but still. The fourth A nucleotide is very unique, and the sixth T nucleotide is also quite unique. So let's mark the second to sixth nucleotide with underlining. And in the end part, there is one A nucleotide, which is relatively distinct, and the final A nucleotide as well. So let's mark these as well. So now again, we zoom in to take a screenshot. Zoom to the 34 entities to get the same quality. Okay, closer to the end. And so we can also add some others from the family as well to make it more comparable. I try to see that all of them are just included once. All right. So it's also important not to activate this screenshot area because it won't be any more a nice picture if you have it disturbing. So screenshot again. File, region, and with some extra margin. So let's take it. Save as PNG. And this was 107, not ITS, but SSU this time. So saved. So SSU for ITS is done. Now we zoom out again and go for the large subunit. Yes, so the large subunit is starting here. And here again, like in SSU, it's a relatively conserved gene, so it is more difficult to find discriminating places for the species. So we have to look carefully. So for sure, one place is here where actually no, one of the other edaphormonas is still has identical sequence like those three edaphormonas livonica on the top. So here it's all shared. Okay, here we see one A, which is unique. But it's always better to have two or more unique ones. So it's better to define this specific micro barcode. Here is a unique T again and a unique C. So that might make a reasonable place to consider. Let's go further. And what I recommend is to try to select this specific barcode in the first 1,000 sequences of large subunit because it's the most broadly used and most variable of this region. So we have moved here. Again, we see a C nucleotide that is unique, and there is another unique C nucleotide here as well. And here we have also length polymorphism, which may be true, maybe not. PacBio makes some mistakes in those homopolymers, so we are not entirely sure if this other species indeed has one less T compared to Edaphomonas livonica. So let's try to look further. Sometimes distinct patterns just strike the eye. Okay, now we are slightly moving outside this targeted first 1000 nucleotides. So let's go back and see some of the places that we initially considered. Okay, so here is also one occasion where we have this polymorphism within our species. This is quite rare in large subunit and small subunit, and most often it is some kind of sequencing error. Okay, so let's try to find some distinctive position. So, still this place looks most appealing, although there is just one difference to other species, or if we can, so if we include this one, it won't help us because it's shared. So let's move back a bit. Here we have also a situation where we have unique C, also nearly unique T here, and another unique T here. So this could be, in fact, our microbarcode. So let's see how long it is. 23. Okay, so a little bit long, but acceptable. Let's still see if there is something more in the left. No. So we can focus on this longer piece. We can also think where to start exactly. Where is the strongest distinction between all? So this is 23. These additional C and A are very constant here. This G is more variable. So maybe let's focus on this part instead. So we can take 25 nucleotides for the mini barcode because it's providing good resolution. The Saccharomyces is not very well alignable here, but here we can see some overlap. Not sure about here though. So the exact position in Saccharomyces cannot be very precisely determined. So now it's also a question whether we want to display other members of the family here. So we can drag a few alignments here to bring them to the picture. Okay, here it's more part of the another. Or let's bring these ones up a bit and drag some extra nucleotides here. We can also bring in something to this and this to fill this line. So now we would take a screenshot that includes this specific area, including the type species, congeners, and also a few members of the same family. But first, let's take a copy of the micro barcode. Again, to Excel file. Now we measure the position of the type sequence to here, position 1. And the same also for position mapping to Saccharomyces. So we can guess that this position is roughly here, although it's certainly not a precise position. Did we go too far? Guess so. Yes. So here, again, replacing all the gaps to 0. And we get confirmation that our mini barcode is 25 nucleotides. The position starts at 542 in type sequence and 521 in Saccharomyces. So let's put it on the map. So that's a large subunit. Since we had other species still relatively close, we allow no mismatches. And the positions were 5, 4, 2 for the legit type, and adding 25 nucleotides is 5, 6, 6. In Saccharomyces, it was 5 to 1 to 5, 4, 5. Okay, the specific nucleotides. Within this species, the best distinguishing is this 4th position T. and the second C in the middle. Fourth position T, and middle C, and also the terminal T. So here we have also information, or we should input information about the relative within species variation and minimum interspecific distance, but I will manually calculate these later. Okay, so let's fill this. Separation from other species of Edaphomonas, because it's not a monospecific group. We delete these other options here. Necessary, ITS2 and LSU. And this I will fill in later. So species level is almost finished, except some extra information in the bottom. So now let's continue to the genus. So it's Edaphomonas. It was proposed by, what was it, Enrica Lara. Since I am the author of the epithet, so I put myself here as well. Let's remove the comment. Type species is Edaphomonas levonica. Tedersoo et Lara. We don't put it into bold. So here we have also those diagnostic nucleotides, but we won't take any screenshots. And we also can define this genus based on the as monophyletic group of different sequences here. But there is one thing we forgot, and this is taking the screenshot for the species and the LSU barcode. So it's better do it immediately, because if we sort those sequences in a different way, then it will be more difficult to get back to the position, and we would waste our time. Yes, so it ends here. Let's do it. File, new region. Let's include with some extra space. Go here. File, save as PNG 107B. LSU, save. All right. Now again we get back to the genus description. So let's go to the bottom, and in this phylogenetic classification concept of the minimum included group, we will have to add all the species that are in this main tree that correspond to both the type species and others of the same genus. So here we have three different entries that we must use for this definition. So we can copy paste them directly from here and delete some information. Monophyletic least inclusive clade in, okay, it's not in eukaryotes, but in the family. In Monadidae family. It encompasses these particular sequences. So we delete this information. And we delete the tabs and line ending marks. This directly refers to this. This 120 was our type species, and in the bottom we actually refer to those extra species in this genus description. So this is for the non-type species. So we can copy them here. Okay, and to make this part complete, let's already add this extra information about this particular species. So these are indicated here, all those three. So let's start from the first one, 626 ending. It's here. It comes from soil in Göttingen, Germany, Pisceabius forest. So we can put forest soil in Göttingen, Germany. The other one, 646, and is here. It comes from New South Wales, Australia, Mittawong village. And it was also soil. Okay, what type of habitat? Woodland. Woodland soil in Mittawong, New South Wales, Australia. And the last one, 627, it's also from New South Wales, Langowan. Let's copy this name, not to make a mistake. And This is rather a forest soil in Langovan, New South Wales, Australia. Okay, so and this comment we just delete. Okay, this is defined, but here we have also some kind of estimate about the species richness. We can do it. I usually would do it later, but I will show one way how it would be. So it would be here simply counting the various groups, and another estimate would be based on the clustering approach using information about interspecific and interspecific differences in the type species. So the type is certainly the first. This is the second, third, fourth, sixth, seven, eight, maybe nine. Let's say 9 maximum, 10 maximum, 11, 12, 13, 14, 15. 15 maximum. So we can put 13 to 15 species. But we need to re-estimate it based on the clustering. So there these values might, the range might change. So here it's again distinguish them from other Monadida based on diagnostic nucleotide signatures. Again, SSU 5.8 titus 2 and LSU. So let's get back to the tree. So again, zoom out reasonably, and again start with the ITS 2 and 5.8s. So I will take those family level stuff to the bottom so that it's easier to compare visually. So let's refine the ITS region. Okay, the 5.8s starts from here. So we can, and this time our target is this group, and then the out group would be all the rest. So if we visually compare, quite surprisingly, we see an interesting distinct area within this before the end of 5.8s. So we can actually think of marking it into this region. So we have here also some variability, which may be true or not. We can't judge. So preferably we leave it out, but if we see what is the distance, so it's exactly 19 nucleotides, and if we want to stick to our 20, we should include one of those erratic positions. So let's see what might be the best solution here. So if we include this G, then this end part seems to be more divergent and hence more informative. So 20 nucleotides will be up to this, from this C here to the G here. If we compare to the outgroup, we see that some of the first 4 nucleotides are matching, 5th to some extent as well, but further it will be quite unique. But there is one group at the level of family which is precisely identical. So actually we can't use this because of its identity. So it seems to be the most close group in this family, so we keep it close also in the alignment. So let's move further to find a distinctive area. So one possibility with quite high conservation of nucleotides is here, but again, at the family level, it's also roughly comparable, so it's not suitable. If we go further, we see another region here. It's 20 nucleotides, and in this other part of the family, there are at least 2 mismatches and 3 nucleotide deletion. So let's see if this deletion persists if we drag it here. Yes, it does, so it's informative. Unfortunately, however, we have here some variability, especially in this one sequence, which varies in 1, 2, 3 different positions. And there is also another one which varies in 2 positions, and yet another one in 2 positions. So it poses some problem because we should either allow more mismatches or add more degenerate positions to the micro barcode. So these features make it weaker. So once more I check. It's 15, and from here it's 20. So we have to define 1, 2, maybe 3 degenerate positions, and allowing 2 mismatches to cover all this genus, but still excluding all the neighboring genus or other parts of the family. So this is what we can at least consider. If we don't have better options, coming in the other part of the alignment. So here the problem is that our genus is relatively variable, whereas the other is not. So we can't use this part here. Here both are variable. Again, it's very difficult to find a common mini barcode. Here, three members of our genus are quite distinct, so we can't make a common barcode here. Distinct also in other parts of the region, which make things more complicated. So here, if we try to realign some part to find differences, we indeed see 1, 2, principally a third one, with the exception of one nucleotide here. And so yes, here principally 3 distinctions, but we must allow at least one mismatch to allow these 2 mutations here. And we should include this first nucleotide here. So it's 3 differences against, at least 3 against other members of the family, and 1 mismatch we can allow. So this makes it a reasonable barcode for the genus. But let's take a quick look further. Again, this problematic group in this genus is not well aligning to the rest, and now here we have reached already large subunit, so we have to keep what we found. So we have to start from the beginning. What is this stretch? This is 22 nucleotides. So if we drag it off, we have here 20 nucleotides. So that's okay. We can select this as the mini barcode. Again, let's map this to the copy this and map it against the type sequence. Saccharomyces is not relevant because ITS2 is not alignable. So we find the start of the ITS2, so it's in this double T. Again, we copy it here. We remove the gaps, and we see that this microbarcode starts at position 269. So let's add it to the template. Okay. Positions 269 to 288, since it's a 20-mer. In type species, Saccharomyces is not relevant, so we delete it from here. And it was ITS2, not 5.8. So instantly, let's also nominate the key distinguishing nucleotides here. So it's the first A, the fourth G, and uh, fourth from the end A. So first, fourth, and 17th. First, fourth, and 17th. So we must allow one mismatch to cover those slightly distinct taxa. For this, we make no screenshots because we are not going to display those as figures. So let's move to the small subunit. So where it's close to the start. What we can do here again is to take members of the same genus up for a closer comparison. So we have to distinguish these from those below. We can see that there is some kind of promise in this batch because the other parts of the family, they are very distinct. One issue here is that there is also small variability in a few positions. So we might look for a better one potentially. So here we see again a similar situation with no internal variability in the genus. So it's actually an excellent region, very difficult to find in SSU in general. And there is actually nothing comparable in other members, other genera of this family. So in a way, it's a perfect microbarcode. So let's define the 20 nucleotides. Okay, this selection is 20. Also, the cerevisia is aligned here. Again, not very well, but still, at least roughly. We copy it here, and we record the distance from the type because it reaches the end. And also to Saccharomyces cerevisiae. Okay, that's the place. Copy and paste. Replace all. So we have position 714 for type and 693 for Saccharomyces. So let's copy. This was small subunit. position 714 to 733 for type, and 693 to 712 in Saccharomyces. And we can underline as all nucleotides as suitable. And in principle, I believe in this scenario, we can allow 2 mismatches. The next will be a large subunit. So here we also see immediately that there are places that are distinct. For example, here, also here. Which is better. It actually doesn't matter. So we can see how much it covers. Okay, so this one is 20 nucleotides. The Saccharomyces is located roughly here. So the closest other taxon, it has 1, 2, 3, 4, 5, 6 mismatches. So again, we are lucky to have such a good macrobarcode. So let's not hesitate, otherwise we will lose it. Okay, first to the Excel table. And then let's map it first according to the type sequence. Excel. And Saccharomyces. Replace caps. And again, we have positions 157 and 155. Let's bring them in. So LSO 157 to 176. And for Saccharomyces 155 to 174. We had plenty of distinguishing nucleotides. So let's define these 2. So certainly it will be the 3rd, 7th, 11th, 12th. Third, 7th, 11th, 12th, and C nucleotide and third G nucleotide. C, third G. We can allow no mismatches, but we can also allow even 2 random mismatches. So principally we have the genus done as well. So next comes the family. Family is Edaphomonadidae. Since I'm describing this, I will be the main author of this family, and as Enrique was involved in descriptions or names, I will put his name here too. Type genus is Edaphomonas. Author name should not be in italics. This should not be in bold. So this is a single family within the entire phylum. We have another phylum members here though, which are not defined further. So we can put the phylum name here. Again, we will have to take care of the micro barcodes as we did it for the genus. When we go up, it will be more and more difficult to find those places. But let's exercise together again, defining those species for the phylogenetic species concept. So we can safely take the sequences representing the genus, because these are present also in the family. And here we see that we have 3 additional sequences at the family level, which we will copy here. Copy it directly from the tree. So we will remove this extra information besides the accession numbers. And these same 3 new accession will be also go below. So we define in notes that this family currently harbors the Edaphomonas genus, but potentially also other genus level groups, which are represented by these extra sequences. So we copy them one by one. We would also add information about their metadata, as we did for the genus, but perhaps let's go through first distinguishing nucleotides at the family level, since this is a bit more difficult. So we open the same window and look for 5.8 and ITS2. Now we have the family. So it's all this group that we have to compare to anything in the bottom, but also Saccharomyces and other eukaryotes. So can we find some good distinguishing region, at least a candidate? So within 5.8, there is something which is distinct from Saccharomyces cerevisiae, but also distinct from other members of this new phylum. So let's first check what is the magnitude of this. It's 14 nucleotides here. In principle, it's fine. The question is if adding anything else is profitable for us. For example, if we add this extra nucleotide on the left, which alternatively contains also C nucleotide, we see that this C is not present in other groups, but the A is present, so it doesn't help us too much. So it's not very useful. If we move to the right side, so there is another T nucleotide here. But this T is also present in other members of the phylum and also Saccharomyces cerevisiae. So this T wouldn't, or this position wouldn't help us either. So we have to consider that in this region we have also two sequences that have one mismatching nucleotide, one of them even in this type species, although it's not part of the type specimen, so we can expect that it's actually a sequencing error, but we can't be sure about the other one. So to be on a safe side, we can include this 14 nucleotide region. Let's copy it from the type. We have to double check it against a larger alignment to see if it's present also in other sequences, other groups which are not displayed here. This is in position seems to be 99 in both Saccharomyces and the type. There are no gaps here. So let's check it up against a bigger alignment. So this alignment is here, and we can copy and paste the sequences here. So what we see is that this sequence is present actually in Amebozoa, also Pusozoa, and of course in all our target. So unfortunately, this one is not suitable as a distinguishing mini barcode for this family. We have to find another one. I was looking for another one previously, and I found something in a small subunit and also large subunit. It took quite a long time because it needs particularly careful screening and comparing. So it seems that, okay, let's copy this also here further for a better comparison. So it seems that in these positions here, it's a relatively good distinction. Plus, there are also some problematic single nucleotide polymorphisms, which we need to consider. And if we select this position here, we certainly have to insert ambiguity K, which would stand for G or T. So we don't necessarily have to do it because it doesn't provide us additional information. And what we can further look is what are the sequences here beyond this intron. Here is yet another distinguishing nucleotide relative to both Saccharomyces and other members of the phylum. So we can definitely include this one as well for better discrimination. So should we select this part, we would end in 23 nucleotides. If we add one more, we get 25 nucleotides, which looks like a more roundish number. So we can copy this to our Excel file, and it makes sense also to compare it immediately against anything else in our very big alignment. So bad news, but it's also found in one amebozoan, but additionally in otherwise it's unique to our family. So it's a pity because the presence in amebozoan is not sufficient. So we should look yet for another one. So here with this, I will conclude. I have to take my son to the training, and those who want to undertake a description of a new species based on DNA or sequence data, you can follow these guidelines. So thank you for attention, and I'm finishing the recording.